Good morning. It's November 29th and this is your daily brief in science. Here's everything you need to know. A fascinating development has emerged from the Gulf of California, where a pod of orcas has displayed remarkable collaborative techniques to hunt whale sharks, the largest fish in the ocean. Marine biologists have documented 4 distinct hunting events from 2018 to 2024, confirming previous anecdotal reports of orcas attacking these massive creatures. The findings, published in the journal Frontiers in Marine Science on November 29, 2024, shed light on the unique hunting strategies of these orcas, particularly their focus on the pelvic area of whale sharks. The orcas specifically target the ventral side of whale sharks, which has reduced muscle and cartilage, allowing them easier access to vital organs. During the hunting process, they work together to flip the whale shark upside down, inducing tonic immobility, which facilitates access to these vulnerable areas. Marine biologist Eric Higuera-Rivas noted that while orcas hunting whale sharks is not surprising, the specific techniques and teamwork displayed are particularly noteworthy. One male orca, nicknamed Moctezuma, was involved in 3 of the 4 observed hunting events, suggesting a potential specialization within the pod. Although researchers did not capture images of the orcas consuming the whale shark's liver, it is believed to be a primary target due to its high-fat content. The study emphasizes the need for sustainable management of marine tourism and activities around the hunting pod to protect their unique behaviors. Moreover, the research indicates that orca pods in the Gulf of California may adopt new hunting techniques, which could be crucial for their survival amid climate change and increasing human activities. The potential loss of whale sharks due to climate change poses a threat to the orca pod's specialized hunting techniques and their overall survival. Francesca Pancaldi from Mexico's Instituto Politécnico Nacional led the study, utilizing photographs and videos from both scientists and the public to identify individual orcas. Recent research from the ICME group at EMBL Heidelberg has shed light on a fascinating aspect of biological regeneration, the ability of organisms to actively redefine their shape during the healing process. Isam ICME, the group leader, stresses the importance of maintaining shape in regeneration, indicating that organisms have an active role in controlling this process. The starlet sea anemone, known scientifically as Nematostella vectensis, serves as a prime example of remarkable regenerative capabilities. This organism not only regrows lost body parts such as its head or foot, but also reshapes its entire body to maintain a consistent overall form. Unlike other regenerative animals that restore lost parts in proportion to their original size, the sea anemone modifies its entire structure in response to injury. Researcher Stephanie Chung observed unexpected cell division in the mouth area of the anemone following foot injuries, suggesting a body-wide signaling response to damage. The extent of body reshaping correlates with the severity of the injury. Minor injuries lead to slight modifications, while severe injuries, like being cut in half, result in significant remodeling. Utilizing spatial transcriptomics and advanced imaging techniques, the research team discovered that injuries trigger molecular changes throughout the anemone, facilitating tissue reorganization and reshaping. They identified active metalloproteases, enzymes that assist in tissue realignment, which were found to be active across the anemone's body during regeneration. The study measured the aspect ratio of the anemone, confirming its ability to restore pre-injury proportions consistently, highlighting the organism's capacity to maintain its original shape. This collaborative research involved teams from the Hubrecht Institute, EMBL Heidelberg, and the German Cancer Research Center, demonstrating a multidisciplinary approach to understanding regeneration. Future research will investigate why maintaining shape is essential for regeneration and how the anemone perceives its own body shape. The findings were published in the journal Developmental Cell on November 29, 2024, under the title Systemic Coordination of Whole Body Tissue Remodeling During Local Regeneration in Sea Anemones. A significant breakthrough in our understanding of protein synthesis has emerged from a collaborative international effort involving researchers from the University of Michigan. This team has successfully imaged the process by which ribosomes recruit to mRNA during its transcription by RNA polymerase, or RNAP. In prokaryotes, such as bacteria, transcription and translation occur simultaneously. This close coordination allows RNAP and ribosomes to work together effectively. The research team has developed a mechanistic framework that illustrates how freshly transcribed mRNAs are brought to ribosomes, effectively bridging the transcription and translation processes. Published in the journal Science, the study reveals that RNAP employs two anchors to recruit ribosomes, ensuring stable protein synthesis. The researchers found that ribosomal protein BS1 enhances the binding efficiency of mRNA to the ribosomal subunit 30S, which facilitates translation. Furthermore, the study identified an alternative pathway for mRNA delivery to ribosomes, facilitated by the coupling transcription factors NUSG and RFAH. Understanding ribosome recruitment is crucial for the development of new antibiotics targeting bacterial protein synthesis pathways. This is especially important as bacteria often evolve resistance to traditional antibiotics. The research highlights the importance of interdisciplinary efforts, with co-leader Albert Weichselbaumer likening the ribosome's recruitment process to a foreperson overseeing construction, ensuring all components are securely fastened before proceeding. The findings, published on November 29, 2024, mark a significant advancement in our understanding of fundamental steps in bacterial protein synthesis. A recent study led by Richard Young at the Whitehead Institute has unveiled a significant issue in chronic diseases known as proteolethargy. This condition is characterized by reduced protein mobility, which adversely affects cellular function and potentially contributes to various chronic ailments. Utilizing single-molecule tracking techniques, the research team found that most proteins examined showed a decrease in mobility by 20 to 35% in disease states. This reduction in protein movement was directly linked to impaired cellular operations, particularly affecting critical processes such as glucose regulation in diabetes through diminished insulin receptor functionality. The study also identified a dysregulated redox environment as a contributing factor to proteolethargy. This environment alters cysteine residues on protein surfaces, restricting their movement and disrupting essential cellular pathways. Increased levels of reactive oxygen species, often triggered by stressors like high sugar or fat levels, were noted as potential causes of this reduced protein mobility. In their experiments, the researchers tested the antioxidant drug N-acetylcysteine, or NAC, which was found to partially restore protein mobility. This provided evidence supporting the role of oxidative stress in the development of proteolethargy. The implications of these findings are substantial, suggesting that around 50% of human proteins could be affected, thereby disrupting multiple cellular pathways associated with chronic diseases. The discovery of proteolethargy as a common feature in chronic diseases could pave the way for revolutionary advancements in drug development. Alessandra Dall'Agnès highlighted the potential for new classes of drugs aimed at restoring protein mobility, which could significantly benefit patients suffering from chronic conditions. The researchers propose that targeting natural pathways regulating redox homeostasis may lead to effective therapeutics for proteolethargy, emphasizing the need for interdisciplinary collaboration to deepen our understanding of the mechanisms driving chronic diseases. A recent study has unveiled promising insights into the role of omega-3 supplements in combating type 2 diabetes and cardiovascular disease. Led by Dr. May Faraj, a professor of nutrition at Université de Montréal, the research indicates that accessible omega-3 supplements could effectively reduce adipose tissue inflammation, particularly in individuals with high levels of low-density lipoproteins, or LDL, often termed bad cholesterol. Published in Scientific Reports, the study highlights a direct correlation between elevated LDL levels and increased inflammation in adipose tissue, which significantly raises the risk for type 2 diabetes. With 529 million people currently affected by this chronic disease worldwide, understanding these mechanisms is crucial. Dr. Faraj's decade-long research has shifted the perspective on LDL, revealing it as a causative factor in type 2 diabetes rather than merely a consequence. The findings suggest that higher blood levels of EPA and DHA, the primary components of omega-3s, are linked to better management of cardiometabolic risk factors. Key results from the study included improved insulin secretion in response to glucose and enhanced clearance of fats after meals, disrupting the connection between LDL-induced inflammation and diabetes risk factors. The clinical study involved 40 healthy volunteers from 2013 to 2019, with 33 participants completing a 12-week regimen of omega-3 supplementation, providing 2.7 grams of EPA and DHA daily, in accordance with Health Canada's guidelines. The significance of this research is further emphasized by its funding from the Canadian Institutes of Health Research and the Canada Foundation for Innovation, marking a substantial contribution to the fields of nutrition and metabolic disease. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.